Welcome to this fascinating journey into the origins of animal life. Today, we are going to explore the evolutionary significance of Ctenophora, commonly known as comb jellies. For decades, we thought we knew exactly how the animal family tree began, but these beautiful, glowing creatures have completely turned our understanding upside down. By the end of this lecture, you will understand why these delicate marine animals might just be our oldest animal ancestors, and how we use mathematics to peer back hundreds of millions of years into the past. Let us dive in. Now that we have introduced our topic, let us think about what it means to build a family tree for all animals. Imagine trying to trace your ancestry back, not just a few generations, but hundreds of millions of years to the very first animal. For a very long time, scientists confidently believed that the simplest animal, the sea sponge, was our oldest cousin. Sponges are incredibly basic. They just sit there and filter water. But recently, a beautiful, glowing jelly-like creature called a comb jelly, or ctenophore, crashed this ancient family reunion. It turns out this creature might actually be the oldest branch of the animal family tree, and understanding it helps us learn how complex traits first appeared. Before we discuss their evolutionary position, let us understand what these creatures actually are. Ctenophores, commonly known as comb jellies, are a unique phylum of marine invertebrates. Unlike the true jellyfish you might be familiar with, they do not sting. Instead, they are characterized by their biradial symmetry and 8 distinct rows of ciliated comb plates. These tiny, hair-like cilia beat in coordination, allowing the animal to swim gracefully through the ocean currents. While they might look simple and delicate on the outside, their internal biology holds secrets that are forcing biologists to rethink the very foundations of zoology. This brings us to the core of their evolutionary significance, their position on the tree of life. Traditionally, sponges were placed at the very base of the metazoan, or animal tree, because they lack true tissues and a nervous system. It made logical sense that the simplest animals came first. However, modern phylogenomic studies, which look at the entire genetic code, suggest that ctenophores might have actually branched off before sponges. This is shocking because ctenophores possess a complex nervous system and muscle-like cells. If they branched off first, it means the earliest animals were more complex than we ever imagined. So, what does this ctenophore first hypothesis actually mean for our understanding of biology? If ctenophores are indeed the oldest animals, it completely rewrites the textbooks we use today. It shatters the old idea that evolution is just a simple, straight line from simple organisms to complex ones. Instead, it shows us that evolution can be quite messy. It suggests 2 mind-bending possibilities. Either complex features like nervous systems evolved more than once independently in different animal groups, or the ancestors of sponges actually had a nervous system and completely lost it over time. Both scenarios radically change how we view evolutionary history. Beyond just rewriting history, this discovery has profound implications for modern science. You might wonder why the evolutionary position of a marine jelly matters to us today. In the real world, studying the unique nervous system of ctenophores could lead to massive breakthroughs in neuroscience. Why? Because ctenophores build their brains and neural networks using entirely different chemical messengers than we do, or any other animal does. By studying an alien-like nervous system that evolved completely independently from our own, scientists can discover new biological mechanisms, which could eventually inspire novel treatments for neurological diseases in humans. Now that we understand the biological importance of ctenophores, let us look at how scientists actually figure out these ancient timelines. How do we know who branched off first millions of years ago? We cannot just look at fossils because soft-bodied creatures like ctenophores rarely fossilize well. They simply dissolve into the ocean. Instead, scientists use a brilliant mathematical concept called the molecular clock. By comparing the DNA sequences of modern ctenophores and other animals, scientists can measure the genetic distance between them. Because DNA mutates at a relatively steady rate, this genetic distance acts like a ticking clock, allowing us to calculate exactly how long ago two species shared a common ancestor. This brings us to the mathematical heart of the molecular clock. The relationship between genetic changes and time is captured by a simple yet powerful equation. D equals 2 times R times T. Let us break down what each symbol means. D represents the genetic distance, which is the total number of mutations or differences in the DNA between two species. The variable R represents the rate of mutation per million years, acting as the ticking speed of our clock. Finally, T represents the time since the two species diverged from their common ancestor. If the genetic distance D increases, it means the species diverged a very long time ago, assuming the mutation rate R stays constant. Let us see this formula in action with a concrete example. Scientists are trying to determine exactly when the first animals appeared on Earth. They compare a specific gene between a modern ctenophore and a modern sponge. They are given a genetic distance, D, of 0.12 mutations per site, and a known mutation rate, R, of 0.0001. We need to find the divergence time, T. We start with our formula, D equals 2RT. We substitute our known values into the equation. Next, we multiply 2 by the mutation rate, giving us 0.0002. Finally, we divide 0.12 by this value. Our final answer is 600 million years. This tells us ctenophores and sponges shared an ancestor deep in the Precambrian era. Let us look at another scenario to understand how scientists find the mutation rate itself. Fossil evidence suggests the common ancestor of ctenophores and cnidarians lived 700 million years ago. Their measured genetic distance is 0.28. We need to calculate the mutation rate R. We start again with D equals 2RT. We plug in 0.28 for D and 700 for T. Multiplying 2 by 700 gives us 1400. To isolate R, we divide 0.28 by 1400. The final answer is 0.0002 mutations per site per million years. This calculated rate can now be used as a baseline to estimate divergence times for other unknown species. Now that we have successfully used the formula in multiple ways, let us understand exactly where it comes from. Why is there a factor of 2 in the equation? Let us set up the derivation. Imagine a common ancestor that splits into two distinct paths, lineage A and lineage B, at some time t in the past. From that exact moment of divergence, both lineages begin to accumulate genetic mutations completely independently of one another. If the mutation rate is R, then the number of mutations that build up in lineage A is simply R times T. Similarly, the number of mutations that build up in lineage B is also R times T. This setup is crucial for understanding the total genetic distance. Continuing our derivation, we now want to find the total genetic distance, d, that we actually measure when we compare the DNA of the two modern species today. This total distance is simply the sum of the mutations that occurred in lineage A plus the mutations that occurred in lineage B. Substituting the expressions we just established, we get d equals r times t plus r times t. Adding these together, we arrive at our final result, d equals 2rt. The intuition here is beautiful. The factor of 2 exists because the genetic distance is growing from both sides of the family tree simultaneously. This proves exactly why our molecular clock formula works. To truly grasp the magnitude of this topic, let us look at this diagram showing the competing phylogenetic tree hypotheses. On the left, we have hypothesis A, the traditional view. Notice the vertical axis represents time, moving from the past at the bottom to the present at the top. In this traditional tree, porifera, or sponges, branch off first, at the very bottom. Now look at hypothesis B on the right, the Ctenophora first model. Here, the very first branch at the base of the tree is Ctenophora. This visual perfectly captures the paradigm shift. It shows how moving just one branch completely changes the evolutionary timeline, forcing us to rethink when and how complex traits like the nervous system first appeared in animal history. We have covered a lot of ground today, so let us summarize our key takeaways. First, we learned that ctenophores, or comb jellies, challenge our basic assumptions and might actually be the earliest branch of the animal kingdom, older even than sea sponges. Second, because they possess complex nervous systems, their evolutionary position suggests that complex traits can evolve independently, proving that evolution is a messy, non-linear process. Finally, we saw how mathematical tools like the molecular clock allow us to peer back hundreds of millions of years, using genetic distance to uncover these ancient family ties. You now have a solid understanding of both the biology and the math behind the origins of animal life. Great job!